Hello everyone, welcome again. Today in this video, I am going to share a new topic related to gram-positive and gram-negative bacterial staining. So, gram staining is a process that is developed by Christian Gram, a Danish physician, in 1884. He developed this technique to distinguish between gram-positive and gram-negative bacteria species. So, basically, on what behalf he did this experiment and how he differentiate between gram-positive and gram-negative bacteria. So, basically, these both bacterial species are totally differentiate on the basis of their cell wall structure, on the chemical and physical property of their cell wall structure. In this video, I'm not going to share differentiation between gram positive and gram negative bacteria. I'm just talking about the technique of gram staining. So, Christian Gram, he firstly staining both the gram positive and gram negative bacteria with a dye that is crystal violet, or we can say gentian violet. So, both gram positive and gram negative bacteria, when stained with this crystal violet dye, then both species retain the stain and they appear purplish or bluish in color. So after staining with the crystal violet, both bacterial species gain the color that is purple or blue. Next step is treatment with iodine. When Christian Graham treated both the bacterial species which are already purplish or bluish in color. After that, he treated both bacterial species with iodine. Iodine is a dye. Iodine is we can say iodine is a solution which makes a dye less soluble. So that crystal violet dye, it adheres less to the cell wall. It adheres to the cell wall because it is less soluble now. And what happened? Again, both gram positive and gram negative bacteria remain purplish or bluish in color. After iodine, or we can say Lugol solution, because iodine solution is also Lugol solution, we can say. After iodine treatment, he treated both the bacterial species with alcohol. Alcohol act as a destaining solution, or we can say decolorizer solution. So alcohol washes away the purple or bluish color which is gained by both the bacteria species. But here is the differentiation between gram positive and gram negative. Alcohol, which act as a decolorizer or dewasher, gram positive bacteria, which whose cell wall is made up of peptidoglycan, because more content of peptidoglycan is present in gram positive bacteria. That's why after treatment with alcohol, the purplish or bluish color, which is due to crystal violet dye, is not washed away. But in case of gram negative bacteria, in gram negative bacteria, where cell wall is made up of more content of lipopolysaccharide, which is more soluble in alcohol. So after treatment with alcohol, gram negative bacteria, lipopolysaccharide cell wall, which contain purple or bluish color, wash away. That's why here is the differentiation. That's why gram negative bacteria become colorless, but gram positive bacteria remain purplish or bluish in color. There is no change in cell wall of gram positive bacteria. Because it retains the stain crystal violet, that's why it is called gram positive. And gram negative bacteria washes away the stain, that's why it is known as gram negative bacteria, and it become colorless now after the staining with alcohol, or because alcohol act as a dewasher, decolorizer, or destaining solution. After that, both the bacterial species again treated with a counter stain, that is safranin. Because gram positive is already purplish or bluish in color, it not retain the stain, that is safranin, that is counter stain. But due to absence of any color in gram negative bacteria again become reddish or pinkish in color after staining with safranin. So gram in the end, gram positive bacteria remain purple or bluish in color, and gram negative bacteria become red or pinkish in color due to the safranin content. So we can say that there are basically four steps. First step is the treatment with crystal violet. After that, both bacterial species become purplish or bluish in color. After that, second step is treatment with iodine or Lugol solution. And the effect of the iodine is that both the cells remain purple or bluish, but the dye, which is crystal violet, become less soluble, and that's why it adheres more to the cell wall of bacterial species. After that, there is next step, that is treatment with alcohol, which act as a decolorizer or destaining destaining solution. It washes away the purplish or bluish color in gram negative bacteria, because in gram negative bacteria there is more content of lipid. That's why alcohol is more soluble in lipid. So gram negative bacteria become colorless, because because lipopolysaccharide which have color, it after the treatment with alcohol becomes soluble in alcohol. That's why all color washes away. But in gram positive bacteria there is a layer of peptidoglycan. That's why alcohol not retained in the gram positive bacteria. That's why alcohol not washes away the color in gram positive bacteria. After the counter staining, that is safranin, which is the last step, gram negative bacteria become red or pinkish in color, and gram positive bacteria become purplish or bluish in color. This was the whole process. There was 4 step. So it is the main process which differentiate between gram positive and gram negative bacteria. So I hope this video will help you in differentiation between the technique, in understanding the technique of gram staining. In the next video I will talk about the differentiation or main points related with gram positive and gram negative bacteria. Thank you, friends.